Okay, guys, so here we are. So, concept 14.2, the second part of the chapter here. Again, this is a fairly long and dense chapter. The information I don't think is particularly difficult, but there's a lot of it. There's a lot of reading in chapter 14 and chapter 15. So, take your time, just go through it one concept at a time, take a break, and then come back to it and review. I would say look at the chapter summaries first, and then perhaps do some reading, and then go watch one of the videos or links online. Come back and do some reading, that type of thing. Vary it up so that you don't get bored and fall asleep and drool on your textbook there, because that is an expensive textbook and you don't want to get spittle on it. All right, concept 14.2, the laws of probability govern Mendelian inheritance. I know this is a biology class and you're probably wishing, wow, I wish we could work some more math into class. Well, you're in luck because we're about to do that. So the laws of probability govern Mendelian inheritance. And it says Mendel's laws of segregation and independent assortment reflect the rules of probability. So remember, the law of segregation says that the alleles for a particular character or trait, they are randomly sorted or assorted, segregated into the various sperm or eggs. And then independent assortment says that the different traits really don't affect one another. As long as these are traits that are either the alleles are located on non-homologous chromosomes, or if they are on the homologous chromosome, the same chromosome, perhaps they are far enough apart that it's unlikely that they're going to be inherited together. That would apply for the independent assortment law. So let's talk about something simple like tossing a coin. For example, when tossing a coin, the outcome of one toss has absolutely no impact on the outcome of the next toss. In the same way, the alleles of one gene segregate into gametes totally independent of another gene's alleles. The probability for all possible outcomes for whatever you're talking about has to be 1. So, for example, if you were flipping a coin, there is a 0.5 chance that it's going to come up heads and a 0.5 chance that it's going to come up tails. So that's going to equal out to 1. And every time you flip the coin, that has no effect from the previous flip. In other words, there's a 50-50 chance every time that it's going to be heads and tails. If you were to flip the coin 9 times in a row and it came up heads 9 times in a row, that is very unlikely, but it could happen. So probability says that is a low probability, but that doesn't mean it's impossible. So if you flip that coin and 9 times in a row it comes up heads, then you flip it the 10th time. Our human nature is to bet tails because it doesn't seem possible that it could come up heads. Now, for it to come up heads 10 times in a row, yes, that is even more unlikely than it is to come up 9 times in a row. But remember, each individual flip is a separate event. So on that 10th flip, there's still a 50-50 chance of heads or tails. The previous 9 flips has no effect. Also, when you're flipping 2 coins, if you were to take a coin in your right and a coin in your left, those 2 flips have nothing to do with each other. So they do not interfere or interact with one another, and that kind of goes to that law of independent assortment that Mendel was talking about with alleles and genes. Oftentimes when you're doing probability or you're working with genetics, you use a Punnett square, which is a visual aid to help you figure out the various percentages. But you can use your basic math skills and the rules of probability to kind of figure this out, even without a Punnett square. Let's talk about 2 of those rules. One of those is the multiplication rule, and one of them is the addition rule. So the multiplication rule means or states that the probability that 2 or more independent events will occur together is the product. Product is a math term that means multiply the answer from the multiplication. So the probability of 2 or more independent events occurring together is the product of their individual probabilities. So let's think about a good example here before we get confused with the words. So let's go back to flipping 2 coins. What's the probability that we will have 2 quarters, one in our left, one in our right hand? We flip them both. What's the probability that they will both come up to be heads at the same time, each on that flip? Well, we know the probability of 1 quarter coming up heads is 1/2, 50%. We know the probability of the other quarter coming up heads is 1/2, 50%. They have no interaction with each other. So 1/2 times 1/2, remember we have to multiply. It's the multiplication rule, and we're trying to achieve the product, which means the answer to the multiplication. 1/2 times 1/2 equals 1/4. So while there is a 50% chance, separately that each quarter will come up heads, there is only a 25% chance that they will both come up heads at the same time. We can use this kind of probability to figure out in an F1 monohybrid cross. So we are crossing two parents that are pure breeding, but one of them is homozygous recessive, one is homozygous dominant. We can use the multiplication rule to figure out various things about the F1 monohybrid cross. And that's because segregation in a heterozygous plant, say you have a big P and a little p, well, that big P is going to go into one sperm, the little p is going to go into another. That segregation is completely random. It's like flipping the coin. Each gamete has a 1/2, 50% chance of carrying the dominant allele and a 1/2 or 50% chance of carrying the recessive. So we can look at our Punnett square, but use kind of these laws of probability to also figure out certain things. So here we have the sperm from the male, which is big R, little r. There's a 50-50 chance that the sperm cell receives the big R or the little r. Same thing with the egg cell, 50-50 chance that the egg cell gets the big R or the little r. So that's 1/2. So we put the 1/2 by each. 1/2 times 1/2, well, there is a 1/4 chance that the offspring ends up with the two big R's. A 1/4 chance of this combination, a 1/4 chance of this, and a 1/4 chance of the homozygous recessive combination. And that makes sense. You've got the four squares, and you have the 4 1/4. What does that equal? Remember, probability has to add up to 1. That does equal 1. 0.25, 0.25, 0.25, 0.25. That is going to equal your 1. That is your 100% chance of having an offspring. So the multiplication rule states that the probability that two or more separate events will occur together, what are the chances that they will happen together? That is the product of their individual probabilities. So again, like flipping a coin, what are the chances that two quarters will come up heads together? Well, one quarter has a 50% chance, the other has a 50% chance. Multiply that together and you come out with a 0.25, 25% chance of that happening together. The next probability rule we're going to look at is called the addition rule. And the addition rule states that the probability that any one of two or more exclusive events will occur is calculated by adding together their individual probabilities. So with this rule, we're calculating what are the chances that of two possibilities that one of these two possibilities will occur, or if there are, say, 6 possibilities, what is the chance that these 4 of these 6 will occur? So think about like rolling dice, rolling one die, and you have 1, 2, 3, 4, 5, 6. Those are your possible options that you will roll. Well, what if you were to ask, what are the chances that I will roll a even number? So a 2, a 4, or a 6. What are the chances that I will roll a 2, a 4, or a 6? Well, the way to figure that out is using the addition rule. If we add up all the individual probabilities, we'll get the probability that we will roll that even number. So the probability of each particular number, 1 through 6, is 1/6, obviously. So all the even numbers, 2, 4, and 6, 2 has a 1 in 6 probability, 4 has a 1 in 6 probability, 6 has a 1 in 6 probability. If we add those together, 1/6 plus 1/6 plus 1/6, we get 3/6, or 1/2. So the chance that we would roll an even number with a single die is 50% chance that we will roll one of the 3 possible even numbers. And that's called the addition rule. We can use that to figure out, for example, in our example that we were looking at, the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. So we'll go back and look at that, but here are the 2 possibilities. Each one of them had a 1/4 chance. Together, that means there's a 50% chance that this would be heterozygous. So let's go back and look at that chart. So out of these 4 possibilities, what is the overall probability that the child will be heterozygous? Well, there's one of the possibilities that's heterozygous. There's a possibility that's heterozygous. If we add 1/4 plus 1/4, we get 1/2. We get 2/4. What if you were to ask, let's say big R is, it shows big R is heads and little r is tails. So what are the chances, we'll say if big R is dominant, that's the only thing you would see. What are the chances, the probabilities that our phenotype is going to be heads? Well, the way to figure that is to add up the 1/4, which would show heads, the 1/4 that would show heads, and the 1/4 that would show heads. That would be 3/4. So the way to figure out the phenotype that we might see is 3/4, 75%. We do that by just adding the separate possibilities or the separate probabilities of these separate events. So the multiplication rule can help tell us what's the probability that two or more independent events will occur at the same time or together. And the addition rule states that the probability of any one of two or more exclusive events will occur can be calculated by adding the separate probabilities together. And the best example again for that is like, what is the chance that you're going to roll an even number off of one roll of the dice? Well, you have the chance of a 2, a 4, or a 6. Each one of those has a 1/6 chance. Add that together, it's 3/6, 50% chance that you'll roll a even number. So we can start to solve some a little bit more complex genetic problems using these rules of probability. We can apply the multiplication and addition rules to start to predict the outcomes of multiple characters or multiple characteristics that we're looking at. So if we want to look at both color of the pea and whether the pea is wrinkled or round, or we want to look at flower color and flower position, we want to look at two traits together, we can start to do that. A dihybrid or multi-character cross is, say, a dihybrid cross. That's the same as two or more independent monohybrid crosses occurring at the same time. So remember, one trait, in this case we're talking about one trait not affecting the other, so we can just consider that separate crosses. So we may be looking at pea color and pea shape together, but they don't affect one another, so we can consider that separate crosses, and then we can multiply to get the information that we want. Say, for example, let's look at a dihybrid cross where we have crossed two parents that are heterozygous for two different traits, and we want to do some calculations. We can do that fairly quickly using our probability rules. So here are our parents right here. This was from our F1 generation. So in our F2 generation, what's the probability of having nothing but the dominant characteristic, two dominant alleles? Well, we could do that using a Punnett square, or we could do that using just our rules of probability. We know the possibility of having the two dominant YYs is only 1/4, and the possibility of having the two dominant RRs is only 1/4. So what's the probability of having YYRR all dominant? Well, we can multiply those two probabilities together, and it's only 1/16. So we could do a big Punnett square that would have a large number of squares in it, and when we calculated that out and looked at it, out of the 16 squares, one of them would have this combination. What about a different probability with a big Y, little y, and two big R's? Well, that is a 1/2 probability of having the big Y, little y, and again, only a 1/4 probability of having the two big R's. Multiply those together, and it's a 1/8 chance. So you can use mathematics instead of your Punnett square to kind of calculate this information as well. So here we're going to use both the law of multiplication and addition to calculate what we want. So here we're using the laws of multiplication to calculate certain probabilities. So the probability of arriving, this is actually a cross with three different characteristics that we're looking at, three different characters. But we know that, again, we're crossing a parent that is big P, big Y, big R, little r, because these are our hybrid crosses that we're talking about here. And so then we could calculate those probabilities for these each individual possibilities. And then down at the bottom, how about this question right here? What is the chance of at least two recessive traits? Out of all of this, what are the chances that the offspring will have at least two recessive traits? Well, we can take a look and add all of these up, because each one of these has at least two recessive traits. Add all that together, and it's 6/16, or a 3/8 chance, a 3 out of 8 probability or chance. So here we're using both multiplication, the rule of multiplication, and the rule of addition, just to calculate various things about the probabilities. And with practice, you can probably get faster using these rules of probability and some simple math than you can drawing out a Punnett square. Some people are more familiar and comfortable with the Punnett squares, and you can do it either way, but you'll find if you practice that you might be able to do it faster just using these basic mathematical rules. And I want to remind you that this information doesn't predict the exact number of offspring. So this may say 6 out of 16, but we can't guarantee that there's going to be 16 offspring or 16 children. This is just a chance or probability. It's kind of like a ratio. The larger our sample size, the more closely we are probably going to match this number. So if we have, going back to our coin flipping model, so if you flip the coin 2 times, now probability says there's a 50% chance every time that it will land heads or tails. But if you only flip the coin 2 times, you're likely to get 2 heads or 2 tails. But if you flip it 100 times, you're much more likely to see that 50-50 ratio or that 50-50 probability. The larger your sample size, the closer to perfect, you might say, or the closer to the actual ratio that you'll see. Because a small sample size or a short time frame can sometimes make it look as if the probabilities are skewed. So that probability is for long time frames or great amounts of data.